Okay, so this is lecture 1, part 2 for advanced physiology. And we were talking about the forces that drive passive transport in the body. Okay, and so we said there were these three forces: concentration gradients, electrical gradients, and hydrostatic pressure. For the rest of this lecture, we're going to focus on concentration gradients and the principles, basically that drive diffusion, which is transport due to concentration gradients. Okay. So, why does concentration gradient drive transport? How does that happen? So the key to why diffusion happens is that molecules. So all the molecules, anywhere in the universe, are constantly in motion. All molecules are moving all the time. This motion is called Brownian motion. Presumably because some person named Brown discovered it. And the key to Brownian motion, this motion that the molecules undergo, is that it's random. Okay, any molecule at any given time is equally likely to go in any direction. Okay, so so this guy's going this way, this guy's going that way, that guy's going that way. In the next moment, all those directions will change randomly. This movement is totally random. Okay, if you were to watch these guys, they'd look like they're just like vibrating and moving all over the place. Okay. Okay, but we just talked about. We're interested in transport, right? We're interested in the movement of molecules from one location to another. And we're saying that that's due to this random movement of molecules. So if molecules are moving randomly, how do they accomplish transport, right? How do they go from one side to the other? Why don't they just move in both directions all the time? Is it that somehow when the concentration is high, they're more likely to go this way than that way? Does that make them not move randomly? And the answer to that is no. So the molecules over here are moving randomly, and the molecules over here are moving randomly. So what causes them to have this net movement from one side to the other? And the answer is has to do with concentration gradients. So if you take a bunch of molecules and cram them together at one place, and make them highly concentrated. So these molecules are still trying to basically move randomly in all directions, but you crammed them all together over here, so they can't really go like this. There's really nowhere for them to go. In the direction of sort of towards the left here. They can't really move to the left any further than they already have. Whereas if they were to be moving to the right, they would have plenty of room to do that. So basically when you cram molecules together, they can't get any closer together, whereas they can move apart. So they're not going to be able to move closer together, but they're going to be able to move closer apart. And if you look over time, what you'll find is that because there just are more opportunities to move away from each other than toward each other, you'll find that there will be a net movement. Overall, molecules will tend to be moving away from one another. So even though individually they're all moving just by chance, there are a lot more opportunities to move away from each other than there are to move toward each other. And so there's going to be a net movement of molecules from the area of high concentration to the area of low concentration. So, and if you've got a membrane here, what you find is that there's just a lot more molecules over here. And so just by chance, there's going to be more opportunities for them to move from high to low than there are molecules going to be going from low to high. So there's going to be movement. The overall movement is going to be from high concentration to low concentration. So let's get more specific about that. Okay, we're really interested in transport across a membrane. Okay, so let's say let's set up a situation where we've got a container of fluid right? So, hello. We've got this container of fluid here, right? So it's full of fluid. There are solutes, these little blue balls in the fluid. And then we've got a membrane that divides the fluid into, let's call it a left side and a right side. And key thing to know is that this membrane that we've put in there is permeable to the solute. So the little blue balls can pass through the membrane, but the membrane divides the two compartments into left and right. And the thing we're going to want to ask is, what's the overall flow of solute molecules? Left to right, right to left, or neither? And when we talk about that flow, the technical term for that is flux. So there's flux of molecules moving from left to right. Call that flux left to right. And that's when one of these guys kind of happens just by chance to move over here, and then he happens by chance to go to the other side. Okay, so that's one-way flux from left to right. Similarly, there's flux from right to left, and that's when one of these guys meanders over here and then happens to go across to the other side, right? So we have flux left to right, we have flux right to left. So, and then what we want to ask is, what's the transport happening in this case, okay? And actually, to know the answer to that, we have to ask the question about the concentration of solute on the left and the right. Okay, so sorry. And the situation we're looking at here is the one we're going to start with first. In the scenario that's been drawn here, the concentration of solute on the right is equal to the concentration of solute on the left. Okay. So basically, the number of solute molecules on the left is equal to the number of solute molecules on the right. Intuitively, you can understand what that means. So how big is the flux left to right? Well, it depends on how many of these little blue balls there are, right? So the more of the little blue balls, the higher chance that one's going to end up at the membrane, and that one's going to go across. So the flux from left to right depends on the concentration on the left. And similarly, the flux from right to left, the more solute molecules there are on the right, the more likely they're going to go from right to left. So the flux from left to right depends on the concentration on the left. Flux from right to left depends on the concentration on the right. In this case, the concentrations on the two sides are equal. And as a result, sorry, hold on a second. Okay, so since the two concentrations are equal, then as we kind of just explained, that the two fluxes, the flux left to right, is going to be equal to the flux right to left. So, and then we want to be able to quantify transport. What we want to do is put a number on the question, what's the overall flow of solute molecules? So we're going to call that net flux. We'll need to pick a direction, so let's just say net flux left to right. What's the overall flux left to right? To calculate that, what you need to know is the flux left to right minus flux right to left. In this case, both flux left to right and flux left to right and flux right to left are equal. So the net flux left to right will be 0. No net flux, no net flow of solute from left to right. we could also just as easily define net flux right to left, which is just going to be the opposite, flux right to left minus flux left to right, and which is equal just to the negative of flux left to right, which, and of course that's also 0 in this case, right? Sorry about it's a little messy here, but you get the idea. Okay, so net flux left to right is 0, net flux right to left is 0. There's no net movement of molecules, no transport. Does this mean that there is no flux whatsoever? Do any molecules ever pass through the membrane? Answer, of course, is yes. Sometimes some of these guys are going to go randomly from left to right. Sometimes some of these guys are going to go randomly from right to left. But the key is that the overall flux is 0. There is no net flux, right? So well, that's no transport. If you were to, so let's make a graph. Showing, so let's say we wanted to say, let's look at the concentration of solute on the left. Over time. What you find is that concentration won't change, right? You have guys moving from left to right, but you have just as many guys moving from right to left. So the concentration of solute in the left chamber will be stable over time. This is what we call equilibrium. Right? Things aren't changing over time. So when concentrations are equal, there's no net flux. The system's at equilibrium. Now, let's contrast that with another situation. Concentration of solute on the left is greater than concentration of solute on the right. So what do we get here? So we get guys going from right to left. We get guys, left to right. We get other guys going from right to left. But because we have many more molecules on the left than on the right, in any just little time window, there's going to be more chance of molecules moving from left over to right. There are just more of them here than there is the chance of moving from right to left. So there's going to be a big flux left to right, a smaller flux right to left. Flux left to right is big, flux right to left is small, and then net flux left to right equals right flux left to right minus flux right to left equals big minus small equals a positive value. So there now will be a positive net flux left to right. We could also define this just in the opposite way. Net flux right to left equals flux right to left minus flux left to right equals small minus big. And now it would be a negative number, right? if you were to say, what's the net flux from right to left, you'd say it's a negative value because there's actually more things going from left to right than there are right to left. And you're just saying the same thing, just defining it differently. Okay? And so here you see that a concentration difference from one chamber to the other generates transport. And that transport goes from high concentration to low concentration. The net flux will be in the direction of high to low. Okay? Going back to our graph, if we let this process continue, say, this was the curve when the solute concentrations are equal. Now, we'll say concentrations are unequal. Solute concentration on the left starts off as being high, but over time, it will decrease because there's net flux from left to right. There's more guys moving from left to right than there are from right to left. So you see that this system is not in equilibrium. The concentration will decline in this case. And depending on, concentration on the right, eventually it will start to level off and will probably eventually reach equilibrium. But initially, you're not at equilibrium. Transport is occurring. Okay? So, transport is highly dependent on, the passive transport is highly dependent on concentration gradients. Okay? And we actually want to be able to, here we said net flux left to right equals 0. Here we said net flux left to right is greater than 0. And we're going to want to get quantitative about this. We're going to want to be able to say, how do we predict what the net flux actually is? What the sign is and how big it is. So basically what we're saying is what is the rate of diffusion through a membrane depend on? And luckily, we actually have an equation that defines that called Fick's law. And there's a lot of ways to define Fick's law. If you look up Fick's law, you'll see lots of variations of it. We're going to define a relatively simple version of it. So net flux left to right is equal to P times A times CO minus CR divided by delta X, where P is, actually I'll start with, so net flux, we know what that means, right? Rate of solute movement across a membrane. The unit is moles per second. A is the surface area of the membrane. So net flux, so A is up here in the numerator, so bigger surface area gives a bigger net flux, and that makes sense, right? You can imagine if there was a bigger chamber, bigger membrane, you'd have a higher number of molecules moving across than if you have smaller chambers with smaller membrane. So area of membrane is important for determining flux. So concentration left, Cl and Cr are concentration of a solute left on the left and right side, right? So concentration difference is a major factor in net flux, of course. Delta X is the width of the membrane. What we mean by that is the actual width here of this membrane. So if you had a really thick membrane, right, and delta X was big, it's harder for material to pass through than it is for a thin membrane. And so delta X is actually in the denominator. Bigger delta X, smaller net flux. Okay, and that's because the wider the membrane, the slower the flux through it. And then the last factor I'll put up here just because it's one of the things that it's one of the numerator guys. P is the permeability of the membrane to the solute. So, and what that means is, how permeable is this membrane to the solute? So we said that, this membrane is basically completely permeable. In this little case study, the membrane was completely permeable to the solute. But of course, that's not always the case, right? I mean, we know that if you look at the plasma membrane, right? So the plasma membrane to the phospholipid bilayer is not at all permeable to sodium. So, if we were doing this calculation for sodium across a phospholipid membrane, it wouldn't matter how big the concentration difference is or any of the other things. If P is 0, net flux will be 0. And the bigger the permeability, the more transport there's going to be. Okay, so let's look at some of these factors a little more. So, first we'll look at concentration difference between left and right. So, concentrations have basically two effects on diffusion. Okay, so the first is that they completely determine the direction of flux. So if concentration on the left is greater than concentration on the right, then net flux left to right is positive, whereas if concentration on the left is less than concentration on the right, net flux left to right is negative, meaning there's more flux from right to left, right? And this is just that old story. Things move from high concentration to low concentration, but here you can see it in the equation. Okay, the sign of this factor, Cl minus Cr, determines the sign of the flux. And then the other thing is that it affects the size of the flux. So if we were to make a graph of net flux versus Cl minus Cr, you'd find that you'd get a line, right? Bigger concentration difference, bigger net flux, and it's what we call a linear effect. If you double the concentration difference, you double the net flux, right? So if this is twice as big, then this gets to be twice as big. And of course, concentration difference is going to be sort of the major factor when we think about diffusion, right? This is what we think of when we think of diffusion, the concentration difference as being the driver. But there are other things that affect it too, as we were talking about. So the surface area, and again, the effect here is linear. So net flux versus A, you get a line. We're going to find that this is going to be really important in the lungs. The surface area of exchange is going to be a really major factor in pulmonary physiology. Delta X, the width of the membrane. So this is the only factor that's in the denominator, meaning that bigger delta X, bigger, wider membrane gives a smaller flux. If we were to graph that, what you'd see is an inverse relationship. It would look something like this. We're going to look at this a little more carefully in the problem-based learning in the lab, but bigger delta X, smaller flux. So as this gets bigger, this gets smaller. Again, this will be actually probably most important in the lungs. The lungs work really hard to keep the membrane between air and blood as thin as possible. And there are a lot of conditions that cause that membrane to get thicker, and that can severely reduce the rate of gas exchange between alveoli and blood. And then permeability. Let's actually go to the next sort of slide, talk about it. So for permeability, again, it's in the numerator here. So you could replace the A with a P, and they get the same effect. Higher P, higher flux. The effect is linear, so doubling of P, you get a doubling of flux. What are some of the things that can affect permeability of a membrane to solute? So let's just kind of make a little list. So hydrophobicity. So let's say sort of permeability, or how about, I don't know, things that affect permeability of cell membranes. Okay, so the hydrophobicity of the solute is important because, as we know, the plasma membrane has a hydrophobic core. So hydrophobic things tend to be a lot more permeable than hydrophilic things. So you may remember from A&P 2 that there are hormones like thyroid hormone that is pretty nonpolar, and it passes quite readily through the cell membrane, is quite permeable to that hormone. Whereas other hormones like insulin are water soluble and they really can't pass into the cell at all. Okay, another factor that is important for permeability is size. So small molecules tend to be more permeable than large molecules. Okay, we'll see that this will be a factor in like pulmonary and respiratory physiology. And renal physiology especially. And then probably the most important one for understanding physiology is the role of membrane proteins. Okay. So, right, sorry, so this slide, I was just showing, anyway, forget about it. Okay, so this slide shows the types of proteins that are important in passive transport. Okay, so we're not looking at pumps right now. Pumps are active transport, and they're also really important, but we're not going to look at them yet. Okay, what we're looking at in this slide are just the proteins that are involved in passive transport. So, just thinking about sort of cell membranes and what can pass through them, right? Things that are hydrophilic, like ions, they just can't pass through the plasma membrane very well at all. They can't pass through the phospholipid bilayer. And similarly, largish molecules, even if they are fairly hydrophobic, if they're very large, they can't pass through. And there are a lot of things. I mean, think about again, going way back to to that first slide, the things that have to pass in and out of cells. Nutrients, things like glucose or fats and proteins, ions, water, hydrogen, H plus. These are all things that just don't pass through the phospholipid bilayer very well, and so really, membrane proteins are very vital for transport of lots of critical things in cells. So let's kind of just look at different types of transport proteins, and that'll finish us up for this first lecture. So when we look at the transport proteins that are involved in passive transport, we find that there are basically two major types. So I'll divide them with this line here. So the ones up here are what we call channel proteins. And this guy down here is a carrier protein. Okay. And the difference is, so channel proteins are kind of donut-shaped proteins. They're proteins that are embedded in the membrane. Right, so here's the phospholipid bilayer. Here's the protein. It's embedded, goes all the way through. It's a transmembrane protein. And this protein has a hole in the middle that things can pass through. It's called a channel, right? So it's a channel protein. It's got a hole for things to pass through. It's called, the pore is the technical name for that hole. And, so that pore lets through things that can't make it through, that can't pass across the phospholipid bilayer. So like ions, there are channel proteins that allow ions to flow through the pore. Glucose, there are channel proteins for glucose. We actually already learned about a channel protein that's really important. It's really present in pretty much all cells in the body, right? Our old friend, the aquaporin, right? Aquaporin is a channel protein whose pore is permeable to water, and it lets water flow through the cell. So, and being just a hole, these guys can't force things to go one way or the other. So the drawing is showing X passing from extracellular space to the cytoplasm, but X could just as easily pass the other way. Okay, the protein can't control that. But there are things that the protein can control. So first of all, a lot of channel proteins are what are called gated. Okay, so that's shown over here. So they'll have a gate that'll either be closed, in which case nothing can pass through, or it will be open, in which case things can pass in or out. and different proteins, what determines whether it's closed versus open. Lots of different things can affect it. So you may remember there are voltage gated channels where there are changes in the cell's voltage can cause the gate to open or close. But there's lots of other things, signals inside the cell. We'll actually learn about a fair number of these examples of these. So that's one thing, is that many of these channels are gated. Not all of them. Another factor that's important about these channel proteins, so you've got this pore and stuff can pass through, but can anything pass through? It turns out in most cases the answer is no. Most channel proteins are selective. That the pore, so this is actually a sort of a detailed structure of a channel protein, and this is the pore here. The pore has a part of the protein shown sort of here and here called the selectivity filter, and the selectivity filter it allows only one or maybe a few types of things to pass through and nothing else. So like let me just get rid of this because it's a little messy, but so like here we have a potassium ion selective channel. This protein has a part that there's a portion of the protein that creates a selectivity filter, and only potassium ions will fit through that filter, nothing else. So this would be a channel protein. It would have a pore, but only potassium ions could go in and out, nothing else. And there are all kinds of different selective channels. There are channels that are selective for pretty much every ion that we know of. Sodium selective channels, chloride selective channels, calcium selective channels. Some channels are somewhat non-selective, so there are channels that have selectivity filters, but those filters will allow, say, sodium or potassium or calcium. Actually, there are some that allow all three. But, they won't let anything else through. They're still selective, but they're not quite as selective. Again, we learned about one, a channel protein that is selective for water. So aquaporins have a selectivity filter that only allows water to pass through. And water can pass through these guys. Sodium can't. And it's a pretty amazing thing. There's actually all kinds of, it's just the structure and the way these pores work is pretty incredible. There was lots of incredible work done to figure out how they work. So this protein can actually pick out water molecules and let them through, and not let things like sodium and potassium through. So again, it's a pore, but it's a selective pore. Okay. So that's channel proteins. Okay, and then we have the other kind of protein that is involved in passive transport across membranes is what is what's called a carrier protein. So a carrier protein does not have a pore. Okay, this is a protein that's a transmembrane protein, but there's not a hole that goes straight through it. Instead, for a solute to make it through a carrier protein, it first has to come in to the protein, and it actually has to bind to a binding site. Okay. So there's this binding site for this little guy, X. X has to come in and bind to the binding site. When it does, the protein changes conformation and opens up at the other end, at which point this little X molecule can exit. Okay? A couple of important points about these kinds of carrier proteins. So the first is that this mechanism is purely passive. The cell doesn't have to use ATP to make any of this happen. Okay, just the binding of X to the binding site causes the conformational change, which allows the X to be pulled through to the other side. So that's one thing, is that it's purely passive. The other thing is that it can actually work in both directions. So there can be X molecules that come in from the inside, bind to the binding site, and the conformation changes in the other way and lets it out. So the two key principles here, it's passive and it works in either direction equally well. Okay, so what do these carrier proteins do? How do they, what is this about? How does it differ from, passage through a carrier protein or just across the membrane. Okay, and so and as you can kind of imagine, it's really pretty different. Okay, so here when we're talking about flow through a pore, I mean this is still just just stuff that happens to be near the membrane can pass through the pore and go to the other side. And it's really fundamentally no different than things that pass directly through. It's just that maybe this is so let's call this let's call this Y. Let's say Y is another molecule that can pass directly through. X is a molecule that has to pass. So say this was like hydrophobic, thyroid hormone, whereas this guy is maybe a sodium ion. But the basic mechanism here is the same whether you're going directly through the membrane or through a pore. And these processes are called simple diffusion. And the key is that a process like this, where things are just passing through a permeable, membrane, simple diffusion is governed by Fick's law. So Fick's law applies to these processes here, passage, sort of the passive flow of material across a membrane. Again, whether or not a protein is involved. So a channel protein, really all it does is it affects P. So P would be for a pure phospholipid bilayer. If we were talking about sodium, P would be 0. But if so this will be my drawing of the phospholipid bilayer. So for a phospholipid bilayer, P is 0. For a phospholipid bilayer with a sodium channel, P is high, but the law, Fick's law, applies in either case. It's just that P is 0 in Fick's law, so you get no net flux, whereas here P is high, so you'd have pretty good net flux. But it's still the same process. It's governed by the same law, Fick's law. Whereas a carrier flow through a carrier protein, it actually is not governed by Fick's law. This is what's called facilitated diffusion. And what's the difference? What is the main thing that distinguishes facilitated diffusion from simple diffusion? And here's the main difference. It's the need for X to bind to the binding site. Okay. And the problem is that there's only so many carrier proteins in the membrane, so there's only so many binding sites available. So here's what happens. Okay, I'm going to draw. Actually, I think I have it ready. Right. So maybe I'll try to draw this. Might be easier if I draw it by hand. So let's say we're going to measure net flux left to right versus the concentration difference left to right. Oops, sorry. Okay. So if we look at simple diffusion, okay, if we're going to look at one of these processes here, either passage directly through the membrane or through a transport protein, what you find is, so when there's no concentration difference, there's no flux, right? So if this is 0, this is positive and this would be negative. When there's no concentration difference, there's no flux. As Cl minus Cr gets bigger, net flux left to right will get bigger in a linear fashion, and vice versa. If Cl minus Cr goes into negative, the net flux left to right would also become negative. You just get a line, right? Zero flux at zero concentration difference, and then positive concentration difference, positive flux, negative concentration difference, negative flux. Sorry, let me. That should not. Oh, damn. That should not be squiggly like that. It should be a nice straight line. So this is what you get with simple diffusion. When you have facilitated diffusion, when you have this binding site that grabs the X and pulls it through, what you find, so still, when there's no concentration difference, there'll be no net flux. Okay, there'll be movement back and forth of X back and forth through this carrier, but if the concentrations are equal, the rate of flow from left to right and right to left will still be equal in facilitated diffusion using a carrier protein, just like it was up here. Okay, so that's going to be the same. But here's where you get some differences. Because we have this binding site that kind of attracts X to help pull it through, what you find is that small concentration differences, you can get big flux. So you tend to get higher flux with facilitated diffusion at low concentrations than you got with simple diffusion. Okay? The little binding site helps pull stuff back and forth. But as the concentrations get higher and higher, you find that this curve for facilitated diffusion actually levels off, and eventually the flux no longer increases. You still get flux, positive flux for positive concentration difference, negative flux for negative concentration difference, but you don't get the increase anymore. After a certain point, as concentration goes higher and higher and higher, flux levels off. This is what we call saturation. Okay, you're filling up all the binding sites. These guys are working as hard as they can to move X through. They're saturated. If you add more molecules of solute, you're not going to be able to get any more flow through those carriers. Okay, so this is the big difference of facilitated diffusion versus simple diffusion. Okay, facilitated diffusion happens at a higher rate for low concentrations, but then it levels off and it saturates at higher concentrations. Okay. Okay, and that'll do it for this part of the lecture.